Hello, friends, and welcome back to the Complete Tech Heads podcast with me, Tom Edwards. This week, I am super excited to be joined by Alex Zhavoronkov, who is founder and CEO at Insilico Medicine and an expert in artificial intelligence for drug discovery and aging research. Since 2014, he's published and co-published over 170 peer-reviewed publications and raised over $400 million in capital. He also has 6 clinical trials for drugs discovered using AI. So very, very excited to talk to Alex. It's a guest that I've been hoping to have on for some time. He's joining me from Hong Kong. And we're going to talk about aging, longevity, and how AI can really disrupt this paradigm that we've been living in since the dawn of man and all of the advances that we're now currently witnessing. So first of all, Alex, thank you so much for joining me. How are you doing?
Happy to be with you, Tom, today. Doing very well. Just came back from WEF, World Economic Forum in Davos, which was amazing.
Brilliant. Cool. Well, I hope you had a good time over there. And I understand they were talking longevity at the event as well. Is that right?
Yes, I had a pleasure to present at the Turning Back the Clock session. So for anybody listening, please Google it, and it was a pretty useful panel.
Awesome. Okay, so look, I want to talk to you about the technology that you're working on. But first of all, if we could just frame the conversation with just the very general question of what exactly is aging? What do we mean when we talk about aging itself?
Sure. So first of all, aging is a very complex multifactorial process comprised of the many biological processes transpiring at the same time. So it's very difficult to pinpoint which one is more important at any given point in time, and it's very difficult to address. Some of those processes were referred to as hallmarks of aging, so the pillars that are kind of driving the aging process. And if you're interested in this topic, just Google hallmarks of aging, but I'll give you a gist. Some of the more established ones are genomic instability. So our DNA mutates over time, and there are multiple alterations that transpire during your lifetime, multiple forms of DNA damage, DNA repair. Telomere attrition, so the ends of our chromosomes get shorter and shorter every time the cell divides. Epigenetic alterations, the small plugs on your DNA that modulate, regulate expression of your genes, shift over time, and we lose the epigenetic memory over time. That's why the cellular reprogramming, where you remethylate some of those sites, is a promising technique nowadays, and a lot of people are talking about it. There is loss of proteostasis, so your protein balance gets dysregulated. There is disabled macrophagy, so microautophagy, where you lose the ability to recycle stuff in your cells, dysregulated nutrient sensing, so your cells lose the ability to properly react to the nutrients around them. There is mitochondrial dysfunction. The tiny energy factories in your cells get dysregulated. Of course, there is a process of cellular senescence in general. That's a very big subject in aging research. So your cells get damaged over time, and those cells that did not become cancer and did not die, they just sit there, do nothing, and excrete toxins into the environment. Those are kind of the cellular retirees, so to speak, not to offend anybody. There is stem cell exhaustion, so you lose stem cells over time. you only have the finite number of those stem cells in your body over time, even though many of them divide and regenerate. However, you lose that capacity over time. There is altered intracellular communication. So cells communicate with each other, and this ability is dysregulated over time. So as cells accumulate damage, and since there is this loss of communication, very often since cells do not talk to each other, there are difficulties in proliferation, in repair, in different cellular and systemic functions. There's chronic inflammation, so we constantly our immune system is attacking our tissues, even though very often you don't need to have those processes, and some of those processes actually result in very substantial diseases, including autoimmune diseases. There is dysbiosis, so the change in balance of your microorganisms, because you're not only made of human cells, you also carry a vast number of bacteria, and changes in those bacteria affect the way you age, the way you smell, and many other functions. So again, when all those processes are combined and intertwined, and dysregulated, we age. So whenever we are trying to address aging, we usually do the hallmarks of aging assessment for a therapy and see which hallmarks the therapy may affect.
Okay.
And when we do it at Insilico, we usually try to look for the therapies that work in both aging and disease at the same time. We do the hallmarks assessment, and that allows us to go after multiple indications and multiple therapies at the same time. I hope it wasn't too long.
No. I mean, it's just it's so interesting that there is so many different interlocking things all happening at the same. You're aging really. We are fighting like a rising tide, right? Like there are so many different things all working against longevity all at the same time. It sounds like.
Exactly. And that's why I encourage every one of your listeners who is in tech to consider getting into this area as fast as possible, because whatever you think is important, this is more important. Because your nature is unfair, and whatever you accumulate during your lifetime, you are going to lose. And the end is never pretty.
If you are interested in extending your own healthy lifespan, then I can't recommend this week's partners enough. DoNotAge.org is a company dedicated to helping people like you and me extend their health span through supplementation. On the website, you can check out all of the different supplements that they sell, accompanied by a whole section on the science behind them. You can read the links on where they've got their research from, check out the team that's working on it, and of course, pick up those all-important supplements for yourself. So go to the site and use the code CTH for a 10% discount. You can also find the link in the description for this show. Yeah, for sure. I mean, anybody who's had an ill aging relative will know that the sharp end of the aging process is not fun. So look, that's a really amazing introduction, and I feel like I could dig into every single one of those. I've had long conversations about senescent cells, for example, on previous episodes, because there's quite a thriving community of longevity enthusiasts now, some of whom I've spoken to on this show. But I want to make sure that we keep timings tight, and I want to talk about the technology itself here. So I understand that we refer to the industry now as AIDD, AI drug discovery. So what exactly is AI doing to help us here? What's the real breakthrough? Is it anything to do with like the similar breakthroughs we've seen in large language models and this kind of level of intelligence, or is it a completely different field?
So of course, large language models do help, but that's usually a piece of the puzzle. And in order to understand where AI can help drug discovery and development, you really need to understand how drugs are discovered and developed. And I can tell you that after 12 years in the field, and after completing, hundreds, if not thousands, of projects for the pharmaceutical companies, and also getting into human clinical trials from scratch myself, I am still learning. So you always feel like a novice in this
Mm,
industry. You always learn something new. But the general framework, if you look at therapeutic drug discovery and development kind of from the eagle's eye view, it consists of several steps. One is disease modeling and target discovery. So you really need to understand why the disease happens and what is driving it, and then try to narrow it down to a specific set of proteins or other forms of targets that we can go after with interventions that are driving this disease or are associated with disease, but if you intervene and if you target those proteins, you would be able to modulate the disease in the direction you want it to go. So to do that, there is more than 1,000 probably ways to identify targets. We've incorporated around 60 target discovery philosophies and methods into a system called PandaOmics that a lot of people use. We also teach a course on disease modeling and target discovery, available for free at insilico.com if you want to get into this field from
okay.
scratch and learn how it's done. Um, but in brief, there are multiple ways to understand why disease happens and identify those targets. So one easy way is to actually read the literature, because usually target discovery and disease modeling transpires in academia, and
Mm,
academics around the world and also within the clinical centers try to hypothesize why diseases happen, observe a lot of patients, construct experimental models, do cell-based research, and publish research papers. And for some diseases, especially in cancer, you would see hundreds, if not thousands, of papers published every day.
mm,
So you would not be able to read through those papers as a human being. It's just you're not capable of processing so much information. And considering the fact that this has been going for a long time, and now many of the studies have conflicting evidence, AI may help quite a bit. And that's actually where the large language models, just because it's
yeah.
language, are the most helpful, especially multimodal large language models that are trained on text and imaging at the same time. So here you would be able to identify targets just by talking to the system and reconstruct pathways, networks, etc. There is another way of doing this, and that's processing biological information coming from patients, from cells, and other sources from animals, and trying to utilize the data types like your genome, your epigenome, methylation, transcriptomics, protein expression, imaging of those cells, all that going into machine learning systems that try to figure out what is driving the disease, understanding the difference between normal tissue and normal cells and tissues and cells afflicted with disease, and looking at how those processes transpire in time. And that's actually where aging research comes in a big way,
Okay.
because you can now train different machine learning systems to predict age, because age is one of the features that is present in every living organism. It's basically like putting time in the context of biology, and biology in the context of time. And if you train deep neural networks and generative systems that can also allow you to generate synthetic biological data with age as a generation condition, allows you to train deep neural networks to understand basic biology. So because all of us change in time from birth to death, a lot of biological processes happen, and sometimes we die not necessarily due to disease. We call it natural causes. You can die of aging. That is why it's very, very important to train deep neural networks to first understand the processes of basic aging, and hopefully also look at those processes through the prism of hallmarks of aging and understand it at the tissue level, at the cellular level, at the systems level. Even psychology can be understood through the using deep neural networks. We published several papers where we trained deep neural networks to predict psychological age and subjective age using surveys. And now you can combine many of those data types in large complex models so that the system understands the relationships between those changes at many, many different levels. And at the end of the day, you try to zoom into a protein target that is driving a disease, preferably that is implicated in aging and disease at the same time. That's kind of my philosophy. I call those targets dual purpose targets.
Okay.
And then AI can, and then you get to the next level. So how do you target it? How do you influence those proteins, right? And that can be achieved by either developing small molecule chemistry. So think of, for those of you who are not familiar with what I'm talking about, think about aspirin or paracetamol, those simple drugs that you can buy in a drugstore. Those are small molecule drugs. And that is actually pretty difficult therapeutic area, therapeutic class, because chemistry is still poorly understood. It's much more difficult to work with chemistry in the context of AI than working with biologics. And that's another way to address the problem. You can design antibodies or nanobodies or peptides or all kinds of antibody drug conjugates to also hook up to this protein target, to bind to this protein target of interest and disable it or modulate it in a specific way. So those are kind of two broad directions where most therapies go. And our company is focusing on small molecules. So internally, we design those small molecular structures that disable those targets. Mostly those are inhibitors. And we also provide software for the design of biologics that many industry players use. And that's in small molecule chemistry, generative systems demonstrated very substantial potential over the past 7 years. So my first paper in generative chemistry was in 2016, where we used generative adversarial networks to design small molecules with the desired properties. Wow. And yeah, so
I
we
mean,
were pretty early in the game.
Sorry, just to very, very quickly jump in there. I hadn't even considered the idea of generative chemistry, which is crazy to me because I feel like I should have. But just the idea that you're presumably creating entirely novel compounds that had never been registered by humanity in the past.
Exactly. So
Crazy. I mean,
previously,
that's like amazing, right? Like, do you ever just stop and think when you're at work on a day-to-day basis, like, wow, this is like we are the future? Does that, or are you just so in the moment that it's just we're just getting on with what we're doing today? Do you have those moments?
oh, I get these moments every day. As a matter
Really?
of fact, those are pretty dangerous moments because, as a CEO of a company, you not only come up with ideas, you also need to learn how to kill those ideas very quickly, right? Because too many ideas and too many aha moments may steer the company in the wrong direction. So I sometimes get, more than 10 ideas every day, and I just try to write some of them down, but most of them I try to kill. That one, when we thought about, okay, well, instead of trying to look for a needle in a haystack and try to identify the molecules from the known chemical space that bind to a specific to your target of interest, to a specific pocket, or get into a specific pocket of your target of interest, and have very specific properties, we can generate molecules from scratch, generate those perfect needles, and then choose from a distribution of those perfect needles. So when that idea came up, and by the way, it came up from the work of Ian Goodfellow, who published with Yoshua Bengio the first concept of generative adversarial networks in 2014, when he was still at the University of Montreal, and then moved to Google and to OpenAI and then back to Google. And we were very inspired by that idea of that you can generate images with desired properties instead of trying to search for those images or try to draw them by hand. And we thought, well, maybe we can teach AI systems to think like a medicinal chemist or better, and start generating molecules with the desired properties, because nature is not great at designing good drugs. You might think it designed all the chemistry in the world, but it did not, right? As a matter of fact, after searching and trial and error of many, many nature-made compounds, you will find just a few that work extremely well. And usually they work for unintended purposes. Those could be like defense mechanisms of some bacteria, or those could come from fungi or some other other sources, but they were not intended to treat human diseases. That's why when we are utilizing the generative chemistry techniques, the chemical space that you are working with is much broader, and you can optimize for desired properties like oral availability, so you can take the drug as a pill instead of injecting. That's actually one of the benefits of utilizing small molecules versus biologics, because biologics you always inject, almost always, with very rare exceptions when you can take oral peptides or something like that. Brain penetration, metabolic stability, bioavailability, and many, many, many other properties. So our first system, when we launched Chemistry 42 in 2020, was able to optimize for 32 properties. And we usually do not just utilize one generative system, we utilize the entire zoo. And then even though the systems are pre-trained, you also need some reinforcement learning to ensure that they learn the properties of the molecule that you want to create, and also the structure of the specific target that you want to disable. But again, that's another second step of drug discovery. So you discover your target, you generate your chemistry. Now what? So now you need to identify which patients to go after, right? And there we can use multiple forms of AI. And here again, large language models are very helpful. We can now search through very large numbers of clinical records to understand what patients are going to respond to the therapy, who is going to benefit the most, and also how to design a clinical study. So what clinical study design is more likely to result in a success. And by the way, we forgot to mention that drug discovery is the area where you fail much more often than you succeed. So even if you start the clinical trials with already known target, known molecule, and get into humans, the probability of success is less than 10%,
Wow.
which
Okay.
is pretty dramatic considering that those clinical studies may cost you hundreds of millions of dollars. And the later you progress, the higher the stakes.
Yeah.
And in preclinical world, you fail 99%. If you are talking about novel target, novel molecule going all the way into humans, it's a very rare event to go from scratch, and usually it takes decades to get there.
And
So
is
far, yeah,
AI, sorry, just to jump in, but is technology improving that rate over time, as well as the number of different, compounds that we can um, generate and targets? Is it improving the success rate over time through seeing things like simulation as well?
very good question. So currently, it feels like it does. Um, our company managed to nominate 17 preclinical candidates, so that's one step before going human clinical.
Hmm.
And we've started 6 clinical trials for 5 programs, which is pretty dramatic. I never expected to be in that position, right, and be able to do it so quickly because we started our own drug discovery only around 2019. Before
Wow.
that, we were developing algorithms and providing software to the market. But then we thought, well,
Wow. So,
yeah,
so 3, 4 years
yeah.
since
So 4
2019.
years,
So that's
and
very
we've, yeah,
fast. That's very fast.
that is
Sure, I
very
mean,
fast.
you're an expert in the field. I'm not, but that to me feels like light speed for medical science. No?
Yeah, comparing to a traditional pharma company or a biotech, that's very fast, and that's
Yeah.
a lot. Never expected to be there, but we also have 2 CEOs in the company. So I am in charge of AI, and Dr. Ran Fong, PhD from Harvard, 11 years at GSK, and then he ran a contract research organization that does those kind of services, drug discovery services for living. He joined me in 2021, and he managed to accelerate dramatically. So
Okay.
he uses our AI to accelerate drug discovery and development. But here I must mention that it's not only AI, but you must need to have infrastructure in place and also a range of contract research organization that performs certain tasks for you in order to be successful. And
Mm.
we also rely very, very heavily on robotics.
Okay,
But again, I
interesting.
wanted to kind of mention that currently the jury is still out because the efficacy, so where you test whether the drug works in a certain disease in humans, is usually tested in phase 2 human clinical trials. So where we test safety and efficacy. And so far, I have not heard of any AI drug discovery company that went from scratch, novel target, novel molecule, all the way to phase 2 complete. So there has not been just enough time. Usually it takes, 6, 7 years to get there. Our company started the journey 4 years ago. Now we're in phase 2, and that phase 2 still needs to produce results.
Okay. Well, very best of luck with that. So look, I'm
Fingers crossed.
yeah, for sure. Fingers crossed for like the whole of humanity as well, right? Not just you, but for all of us. If this, I mean, the promise of this technology for me, from the people I speak to, just seems insanely massive. Like, that for you, the more you speak to people who understand this stuff, the more hopeful and optimistic and excited you become, in my opinion. And certainly, this conversation with you has reinforced that. So, I am aware of your time. I'm aware you're an extremely busy man and you have many important things to be doing. So, I will just start to wrap up the conversation by saying, like, what if there were one thing, one sort of area of your research or anybody else's research in this field at the moment that you're most excited about, that you think holds the most potential for kind of transformative technology, what would that be? What are you most excited about in the world right now?
Sure. Well, I'm mostly excited about multimodal transformers for aging research and drug discovery.
Okay.
So multimodality, I think, is the name of the game for this year and maybe the next few years. And I'm not talking about multimodal. It's one single model that can take in multiple data types. For example, text, gene expression, protein expression, methylation, um, histone modifications, imaging data from humans, and in humans from different organs, and also in animals from different organs, and take the drug information and the information about the different response types from different cells and tissues when they are exposed to different drugs, and also clinical information, in order to predict age, generate synthetic data with age as a generation condition, identify protein targets, identify pathways, predict those, reconstruct multiple pathways, generate small molecules, and preferably also do experimental planning and work as a biomarker for age and disease at the same time. So we are working on something like that for a while now. You can see some very early birds that we've published early last year called Precious 1 GPT. Now we're already at Precious 3 and Precious 4 GPT, where we haven't published that, but you can sense the general direction. Precious is, coming from Lord of the Rings, one flock to bind them.
Okay.
And
I get it.
combine
Yeah.
them. Yeah,
Okay.
that's the
Excellent.
idea.
Very good.
And hopefully we'll be able to also integrate it with our fully robotics lab. We have a lab, 6 interconnected rooms run by robots, where you can just throw the sample and the sample will get analyzed. You generate a lot of different omics data types and imaging, and then you expose that sample to a variety of different chemicals and see how it responds, and see whether the system did well or poorly, and reward or punish the system based on what it generated in terms of hypothesis and which compound it picked. And you constantly train, but also you discover novel targets. So currently it utilizes multiple different forms of AI. We want it to be run by one single multimodal system. And I think that's what I'm mostly excited about right now.
Okay, amazing. Well, look, the room full of robots, the multimodality, the dual purpose sites, it all sounds insanely exciting. And I feel like I could sit here and talk to you all day, because any one of the different things, I think I could probably do an hour podcast on. Alas, we need to wrap up, but it's been amazing. So listen, thank you so much for coming on. It's been wonderful to meet you, and I look forward to hearing about all of your exciting progress in the future.
Thank you, Tom. And just as a kind of concluding remarks, since some of your listeners were already exposed to the concept of cellular senescence, I just wanted to mention that probably the number one person in cellular senescence who characterized the senescence-associated secretory phenotype, identified a range of senolytics and xenomorphics, did fundamental work in cellular senescence, has passed away. Her name is Judith Campisi, Professor Judy Campisi at the Buck Institute. So rest in peace, and please make sure that you Google her research, look at her papers, and pay your respects, because that's the person who characterized the entire field.
Okay, Judith Campisi. Amazing. I very much will do so. And as a final note, any awesome AI developers who are looking up Judith or looking up yourself or wanting to work in this field can, I'm sure, find you simply by Googling your name or Insilico Medicine.
Perfect. Yes, Google Scholar is open, so you can look up our recent papers and consider um, getting into the game, because that's the most important game to play.
100%. Brilliant. Well, look, thanks so much for joining me. It's been great.
Thank you. Thank you, Tom.